Hi, good morning. Yeah, I'm sorry to anyone if I offended you by claiming that I wasn't well dressed. I was referring to everything else except the shirts. We're wearing parrots today. Definitely wardrobe by Nicola. Yeah, and honestly, yeah, she's the one thing keeping my look together. All right, here we go. First question, and we're still digging out from under the ones before we took that break. Let's see. It's from Hazel. Good morning, Hazel. Hi again from sunny UK. Can you give your thoughts on this article regarding T cells? And it's one of many I've seen now giving hope for immunity even when antibodies aren't there. Yeah. And could this be an explanation why we aren't seeing reinfections yet? Yeah. I'll add a yet in there. Yeah, totally. Okay, so the article is talking about T cells. It's singing their praises. Thing is, there are a whole bunch of different kinds of T cells, and you need a far better immunologist than me to even go through all the types. So I can bring you this little baby, like egg of an idea of who the big T cell players are and what they're doing. So, broadly, you're going to have your helper T cells that are going to interact with other kinds of cells, and they're going to give them signals that sort of reset them and get them to keep going on. Otherwise, these cells kind of, I don't know, run out of steam and basically just die. Yeah, which is fine. You don't want a full on immune response to go forever, but the helpers are there to keep at least parts of it going when they seem to be doing well, and when there's still something left to do, when there's still antigen left to fight, basically. And so, of those, you've got your helper ones and your helper twos, and there's helper 17s, and just put those aside for a minute. um, the TH1s, T helper ones, those are going to be turned on early in infection, both in humans and now there was a paper that just came out that showed that it's the same sort of thing in monkeys. And so, yeah, these are going to do a lot of work. They're going to start getting you making B cells. They're going to be connected with a killer T cell response, which is another one. Beyond that, they're going to be memory T cells and regulatory T cells. Oh my gosh, yeah. So the reason why we talk about antibodies is because antibodies are easy to measure. And you can measure antibodies just from a little drop of blood. T cells are individual little fighters in your immune system. And to test them, you have to pull out, thousands to millions of them and then show them something. And if the thing that you show them is the thing that they have grown up learning to recognize, then they will basically attack it. They'll start to make signals like interferon gamma for some of these things. Yeah, all of which is just words, right? So let's back this up. Yeah, T cells are definitely important. They are another component of the immune system. There are lots of different kinds doing lots of different jobs that basically end up pushing you in the right direction most of the time. Yeah, there are certain subsets that may be, I don't know, less helpful later on, like the Th2 subset seems to be there when you're kind of at the peak of disease, and I am not sure if they're helping or hurting for the most part. But then yeah, all of these cell types that we've been talking about, even macrophages, they're all different versions of monocytes, which are like their parents, and macrophages. And there are regulatory versions, there are, little fighter versions, and they have all different proteins on the surface and they can do different stuff. So, yeah, antibodies are one dimension out of, I don't know, 10 or 20 of the immune system, and it totally makes sense that somebody somewhere, would be able to make a response, knock out the virus without having any antibody. Just as you can probably win a game of chess without moving a particular piece, if you work at it. Yeah, it's not necessarily the way you would plan things, but yeah, it's a thing that if it happens is not necessarily the end. So yeah, I guess the problem is that even the T cell responses do seem to drop about in concert with the antibody responses. So yeah, while there's more to the immune system than you can see from an antibody test, there's not a good indication that any part of it is going to be particularly durable. But the thing with T cells is, if there's one of those hiding somewhere in your body, just somewhere, hanging out, and it is basically, it has been educated, it is a veteran of a fight against SARS coronavirus 2. There are pathways that can wake that up and cause it to divide really quickly, and all of a sudden you have millions of copies of that T cell that can come in and give you a much faster response to a new infection. So, yeah, they can work. The other thing we've seen from animal models is that in at least the ferret model, and I think one other model, but I can't remember which one off the top, the animals can get the virus a second time, but the second time they'll get fewer symptoms and a milder disease. Now, that's not a guarantee that that's going to happen in everybody, but it is a way that vaccines that don't do everything can still do something. Yeah, so that's like a more of a non-answer than you ever wanted, I'm sure. Yeah, but there you go. That's the little bit that I can articulate anyway. Yeah, so T cells, they're not bad. Yeah, they're actually probably as important, maybe slightly more important, but there aren't that many of them, and you have to pull them out one at a time and kind of test them out in a little Olympics to figure out what they do. And that's hard and super expensive, and so that's not the kind of test that they can just do for anybody. You need your own technician and your own lab, and then, yeah, give them a week and they can run the assays, I think. So thanks very much. This has been Ask Dr. Ben.